We focus on two areas. First, letting our community know about the program and its importance for precision medicine. And second, informing researchers and students about the Research Hub. Today, I am honored to introduce you to Dr. Michael Lyons. Dr. Lyons is the scientific project manager for Vanderbilt University Medical Center, and is a member of the outreach team of the All of Us program's Data and Research Center. He received his PhD in immunology and microbiology from Stanford University, and is interested in applying his scientific background and knowledge in a transnational setting that will benefit patients. As part of All of Us, Dr. Lyons focuses on helping researchers utilize the diverse resources of the All of Us program for their own research needs. Additionally, he develops beginner training materials aimed to broaden the audience of researchers able to engage with the All of Us datasets and expand the impact the program can have on personalized healthcare. Welcome, Dr. Lyons. The floor is yours.
Yeah, thank you. Thank you very much for that introduction, Brenda. So yeah, I will be going over kind of what the All of Us program is and how to use it. So I'll get started. So hopefully you all can see my screen now and see the presentation. Am I good? Excellent. Yeah, so I won't waste time introducing myself again. But throughout this kind of presentation, I'm also joined by several of my colleagues at the Vanderbilt University Medical Center, which is part of the Data and Research Center for All of Us. So if you have any questions, feel free to put them into the chat as I'm kind of going along. I probably won't be monitoring chat, but I'll have several of my colleagues there to answer them. And then we'll also have time for a Q&A session at the end. So we really want to make this as useful for you as possible. So please feel free to throw questions in the chat or ask at the end. So just kind of give an overview of what we're going to be going over today. It's really going to be focused on understanding the All of Us program, and then how to use the data set for conducting research studies. So you can kind of go out, get excited about what we have to offer, and jump in there and start doing some really cool work. Before I jump into the presentation, there's a lot of different communities and people to thank. So without our different participants and community partners, we would not be able to have this data set. So as you can see, there's a lot of different organizations that we work with to encourage participant participation. And in addition to that, we also have a huge amount of different organizations that are part of our consortium in handling the data, analyzing the data, and making that data available to researchers like yourself. These are different participant centers, healthcare provider organizations, and then in the bottom middle, you can see the Data Research Center, which Vanderbilt, which I'm part of, is a part of. So kind of just start us off going into what is the All of Us Research Program. So this is a massive NIH program, and the concept behind it is to develop a database that researchers can engage with that will have a huge emphasis on historically underrepresented populations and communities. So this is going to be a data set that combines a variety of different data types, from biological factors and social determinants, such as genetics, genetic information, electronic health records, as well as different surveys that participants are able to fill out that will kind of give us information on their social determinants of health, as well as kind of health history, social economic status, race, ethnicity. So kind of being able to combine all this different information in one massive data set to get a holistic view of our participants and understand the different things and issues that face them and influence their different healthcare outcomes. One thing that's also really awesome is that we're not trying to take a snapshot and have an individual time point. We're trying to have a longitudinal data set, including all of these different data types, and that will kind of grow with our participants over time so we can see how it all progresses, which will be very, very rich resource to for researchers to go into and access and analyze. So it's something that we're very excited about and proud of. Our overall goal is to get up to and beyond 1 million participants. Currently, you can see that we have almost 500,000 participants signed up and consented to be part of the All of Us research program. And almost 400,000 have completed the initial steps. So that's a couple of initial surveys that we have. And then once they have those surveys filled out, then we can get that survey data going, and then get that data available to researchers like yourself. So you can see that over time, we have kind of hit in the middle here. This is where COVID hit and we kind of leveled off, but we're getting right back on track and going forward strongly to get to that million and beyond. So we're very excited about that. The thing that really does set our data set apart, besides just the overall sheer number of it and the different data types that we're looking at, is that we have a major emphasis on historically underrepresented communities and populations in our data set. So if we're just looking at race and ethnicity, only about 50% of our data set is white, which historically a lot of these types of data sets are of kind of white European descent. So we have a huge emphasis on almost 20% is Black African American, 16% is Hispanic, Latino, or Spanish. And you can see that there's other races and ethnicities included in this as well. So that's something that we're very proud of, and is a major goal of ours, is to keep that type and preserve that. In addition to just focusing on race and ethnicity, we have other categories of underrepresented in biomedical research that we also try to focus and engage with. So one thing, what we're very proud of is that of our data set, 77% of our participants fall under the category of at least one underrepresented in biomedical research category. And you can see how those are broken down over on the chart on the left. So this is for race and ethnicity, age older than 65, education, so less than a high school degree, income, so less than 25,000, and then sexual, gender, and minorities, as well as rural locations. So this is just a very rich data set to look at how populations that historically don't have this type of data set for researchers to engage, which is really, really exciting and awesome. We also don't want to focus on a very specific portion of the United States. We want this to be across the United States. So obviously there will be hotspots because there are some states and places that are more populated than others, but that is an emphasis of this program as well, to have gather participant information from different locations across the United States and see how that impacts different healthcare outcomes. Going into how that data is actually handled and gets to our researchers. So all of this data is collected from our data sources. So healthcare organizations, healthcare provider organizations, self-reported surveys from participants. That's all ingested and taken in, harmonized and refined from the data research centers, and then develops into a curated data repository. And then this curated data set is what becomes available to researchers. So we have about 2 times a year is when we have updates to our database for researchers to go into. And then that will be continuously updating. And we're historically had about 2 times a year these updates, and it's probably moving more to about 1 time a year as we get larger and larger data sets. And that also makes it a little bit easier for researchers to work with, so they're not constantly trying to update their system and research study to incorporate new data. But we are always growing, which is very, very exciting. To actually look at what we have currently, our last major update was in June, and currently available on for researchers to work with row level data is from over 370,000 participants. 74% of this data is from participants that are from underrepresented communities, and 44% of that are from racial and ethnic minority groups. So just a very novel and rich data set to work with. When we actually look specifically at the data types that we have available, we kind of break it down into 6 major categories. So we have survey responses. So these are surveys that participants fill out themselves that look at different aspects of their life. We have 3 basic surveys, which is kind of the initial steps to actually being involved in the program. So that's why we have the highest number for survey responses. That's kind of the barrier of entry. And then they can choose to connect other data types as well, such as physical measurements, which would be height, weight, things like that, electronic health records, And then as well, they can contribute biosamples and blood to get their genomic data linked as well, which can come in genotyping arrays as well as whole genome sequences. And then we also have expanded to include Fitbit records to look at how different measurements and personalized measurements can be impacting personalized health as well. So in our latest release this past year, we also released the genomics, which we're very, very excited about. So this is a way that you can look at participants and link a participant's genomic information to all the different other types of information that we have. So if you wanted to look at linking the genomic information as well as electronic health records, survey data, and physical measurements, you would have a pool of over 81,000 participants to look at. And that number will only increase as we continue to expand our database with future releases. In addition to expanding with future releases for the sheer number, we're also continuously looking to evolve what data types we're looking at. So we're looking at getting more wearable data, more surveys, ancillary study data. So one thing that we're looking at expanding to is microbiome data. So all sorts of different data sources to just continue to get as holistic of a view and picture of participants and things that impact their health as we can. So very rich, and we're continuing to expand. So hopefully I kind of got you all excited about what the data is, and now we'll kind of go into how do you get into the data, how do you access it. So we have a public website, our research hub, and this is kind of where you'll go for your first steps in registering, accessing the data, looking at the data, and then from there you'll be able to kind of become a registered user and then actually work with row level data. And we'll kind of go through what that looks like. But first, we have a very strong commitment to two major things in our program, and that is to have this data available for researchers to be useful, to actually make a positive impact. And then the other is to our participants as well, of keeping this data safe and private, since this is very sensitive and we want to keep that secure. So balancing that is kind of what our major aim is to do. And one way that we're doing that is by kind of developing different tiers of access to the data. So our first and most basic tier is the public tier. So this anyone can access. It's on our data browser website. And this is where you'll be able to see kind of an overview of the different data types that we have, specific numbers on different conditions, different race and ethnicity breakdowns. And all these numbers are rounded to the nearest 20 to not give very specific numbers. And there is no row level data at this location. To actually access the row level data and to start doing like some serious analysis, you have to become a registered user, which is a short, about 2-hour kind of sign off and registration process where you kind of go through training on how to effectively and safely handle and not accidentally download or share this information. And then once you are a registered user, you'll get access to our researcher workbench, which is our cloud computing platform, where you'll be able to go in and actually work with the data. So in our registered tier, this is where you'll actually be able to see the row level data for surveys, electronic health records, physical measurements, things of that nature. And there's also an additional tier within that kind of once you're a registered user, which is the controlled tier. So this has slightly more sensitive material, such as genomic information, as well as a more detailed location. So for registered tier, you get information up to a state level for what state a participant's from, while in a controlled tier environment, you get up to a 3-digit zip code. So the only difference between signing up for these is it kind of depends on what agreement your institution has with the All of Us program. And then you will either have a registered tier agreement or a controlled tier agreement. And then the controlled tier agreement, if you are working with the controlled tier, you'll have like slightly longer kind of security training to go through, but not too much. Both are very, very useful, and both you can do some great analysis with. For kind of having a user get access to this, we follow what's called a data passport access model. So we approve the user and not the project itself. So once you register, you're able to get in there and work with all the data that you want. You'll kind of make it into workspaces, and we'll get into how that works a little bit later. but we are not approving you to look at specifically only this type of data. You can look at all the data, do as many research projects as you would like, and we just kind of approve you to access that data, which is great. We currently have 380 institutions that have an agreement with us to work with our data, and over 2,700 registered researchers that are kind of going through making workspaces and using our data. So we're trying to continue to grow this, make this as useful as a data set as possible, so then we can make some great research and make some advances in personalized medicine. So it's very, very exciting. We're excited to have you all. And so now I'm kind of going to switch over from understanding what the program is, what we do, what we provide, to how do you actually start using this data and work and develop a research project. So to do that, I'm going to move us actually over to our public site that I was just talking about. So this is our public site, our research hub. And on here, you'll find a number of really, really useful information. So you'll be able to see in this section, the data snapshots. Excuse me. So this is looking specifically at this point of time, where is the All of Us research program in terms of participants? What's that data breakdown? So we have almost 340,000 electronic health record participant links, over 400,000 biosamples received. So that means that that will be turned into genomic data once we start analyzing it and getting it in and ingesting it. And we can also look at the latest spotlights. So these are different research studies that are using the All of Us research program data and publishing on that. So we're kind of spotlighting what people are currently doing. Here is a really interesting thing. So this is our research projects directory. So we can explore this. And what this is actually a list of all of the research projects currently ongoing within and utilizing the All of Us data. So part of one thing that we do to keep the researchers and research that's done with our data accountable and make sure that it's not either stigmatizing or being used in a negative fashion is that all people's workspaces and kind of research projects are displayed publicly. So you'll see here, you can go in here and you can search different project titles, keywords, if there's a specific type of project or focus that you're interested in. So if it was breast cancer, you can type in breast cancer and you can see how many projects and what projects are being done with breast cancer. So we do not prevent people from going in and doing the same type of research as someone else, as I was kind of mentioning with that data passport model that we have. So this is a really, really great way to go in, find out what are people looking at, what are people doing, and then you can go through and actually look at what are the scientific questions or the research project purposes, approaches, as well as who's doing that. And this is a great way to find potential collaborators, or if you want to be a little bit more secretive, who's your competition. So this is a really, really great way of kind of going in and understanding that. If we go back to kind of our overall, we can continue going. And a very, very useful thing on starting you on your research journey is looking at our data browser. So if we go to our data browser, this will be what I mentioned in that public tier of like looking at what data is actually available in the All of Us database, because it's very, very extensive, but is not all encompassing. So we might not have absolutely everything you're looking for, but this is where you can find out what you can actually work with. So as you can see here, we have the data broken down into our different categories. So on top, we have our EHR domains, which are electronic health record domains. So this is where we can see our different conditions, drug exposures, lab measurements, procedures. Below that, we have our genomic data. So you can search for different genomic variants, as well as our physical measurement and wearable data. So looking at height, weight, as well as our Fitbit measurements. So if we're interested specifically in testing to see if we're a specific condition, so if we're continuing on with breast cancer, we can actually do a keyword search here and just type in breast cancer and see what type of data surrounding the condition of breast cancer is within the All of Us database. So our top hit here is malignant tumor of the breast, and we can actually scroll over that and see that we have 8,340 participants. As I mentioned, This number in the public database is rounded to the nearest 20. So it isn't necessarily exactly that number. And to get exactly what number it is, you'll have to become a registered user and look in the the researcher workbench, which we'll go to shortly after this. So this is a great way to see what type of data is available and search for specific conditions, drug exposures, whatever your research idea is, what data is available under that category. The genomic variant search works a little bit differently, but is also great. So we have another search bar that you can use, and you can look and search by 4 different ways, methods. You can either search the gene name, the variant number and mutation, RS number, or genomic region. So if we're interested in BRCA1 or BRCA2, for example, so we'll just type in BRCA2 and see what pops up once it loads. So then if we kind of build out this variant table here, and this is what you can see. You can see the variant ID, and then there's also these different, these different columns as well, which will go over protein change, the significance based on ClinVar, the allele count, allele number, and frequency. And then additionally, if we go to something that has a higher allele count, and then go and specifically look at this variant, we can also get an overview breakdown of the demographics of that. So what populations is this more frequent in, and things of that nature. So it's a really, really useful tool of just understanding, okay, what's available, what can we work with? Finally, I want to bring up how to actually go through and then register. So if you're not a registered user, but you would like to be, you can click on this register button, and this will kind of take you through the process of becoming a registered user. So you'll kind of go through, make sure that your institution has an agreement. I believe that yours does. And then you'll go through kind of the training process, as I mentioned, verification of ID, and then this is the training, and then the signing the code of conduct. Then you'll be able to actually access the researcher workbench to actually work with the row level data. So if we actually move over to our researcher workbench, so this is kind of the home page of our researcher workbench, and this is where, as a registered user, you will be able to go and actually start working with row level data. It is a massive resource, and it can be very intimidating. And it takes a little bit of time to work around and get familiar with. So definitely the best thing to do is just kind of get on there, jump on there, get familiar, spend time on it. But I'm going to do my best to give you guys a heads up on what to look for, how to navigate, and how to kind of get started on your journey. So first of all, the way that projects are essentially organized and broken down is in workspaces. So workspaces is where you'll actually be building out, gathering your data and analyzing your data. So when you develop a new workspace, you'll title it with whatever your project title is. And then this is where you're actually going to fill out that description of your work that is going to be on our public website, as I showed earlier, an example of. So this will just be a short kind of process of filling out what are the scientific questions that you're looking at, what are the approaches, what kind of data sets are you using, any hesitation on stigmatizing research, and then this will all be presented to the public on our public data site for everyone else to kind of see. And that's also how we kind of keep it accountable to see if there is stigmatizing research. If we get any complaints, or if there's anyone saying, hey, I don't know if this research is actually beneficial, it might be kind of stigmatizing. That's kind of how we review and make sure that everything is in a positive manner and using our data well. So once we actually develop a workspace, I'm not going to fill out a new one in the kind of management of time. We can actually just kind of use one that I have as an example. This is what you'll actually look at and see when you open up a new workspace. So there'll be 3 main tabs on the top: data, analysis, and about. About will just be describing the workspace, so what you had just previously filled out. So really the first one that you're going to want to look at is the data tab. Data tab is where you're going to actually be going into and interacting with our database to pull out the data that you want. This overall process is that you're going to be starting with all of us participants. So all of a participant entire population, that over 370,000 group of individuals at this moment. Then you're going to be pulling out the IDs associated with specifically the population that you want to look at. So if we continue our breast cancer example, if you're specifically interested in participants that have a breast cancer condition or even a subpopulation of a breast cancer condition, then we will go in, pull those out, and then we will specifically look in the data set builder how to get the data that we want from that population. So if we want demographic data, if we want data on their medications and labs, this is kind of where we're going to do that. And then that will be put into a Jupyter notebook. And that will be the actual environment and notebook that you're going to be used to actually code on, do your analysis. And we'll go through all of this process more in detail, but that's kind of the general overview. So to start, as I mentioned, we're going to be kind of condensing from the giant group of all participants into just that participant group that we want. So if we go to our cohort builder, we'll have essentially a really nice point and click tool system that we can use. So this is where you'll add criteria to develop your actual cohort. So this is where you can look at our different data types. So our demographics from surveys, physical measurements, Fitbit data, genomic sequences, as well as our different electronic health record domains, so different conditions, procedures, drugs, lab measurements, etc. So continuing on our example, if we're looking at specifically breast cancer participants, we can go to the conditions tab under electronic health records. So these are a huge amount of conditions found across electronic health records. And then you can also do another search function as well using this. So this is all organized in the OMOP data dictionary. So you can search through SNOMED IDs, ICD-9 codes, ICD-10 codes. And if you're not as familiar with working with electronic health record information and kind of that coding definitions, then you can also just type in based on a general term. So if we're interested in breast cancer, we can just type that in and then we'll get breast cancer related conditions. So you can see here, there's a number of different conditions that are associated with breast cancer, and how that all kind of breaks down what's within it. You can actually look at the hierarchy here. So you can get kind of more and more breakdown of the different conditions if you're interested in something very specific. Because this is a public and recorded process, I'm not going to be going into there because I don't want to go into smaller number counts. But so if we're interested specifically in the malignant tumor of breast, we can click on that add button. And then this will go to kind of our box here. And we can potentially, if we wanted to, apply modifiers for this event. We can do different age, occurrence dates, visit types. But if you're just interested in looking at participants overall that have had this condition in their electronic health records, you can just click that and hit save criteria. And then from here, you can continue to modify and refine your cohort. So you can either have either or conditions. So if we wanted to have participants that have either breast cancer or different type of cancer, we could add that type of criteria here. So we can go back to conditions, type in again some other type of cancer, lung cancer, if you're interested in that. And then you would have a cohort that has either breast cancer or lung cancer. If we're interested in adding a specific category as well, we can also do that. So if we're interested in linking this breast cancer diagnosis to genomic data, we can do and group 2 and go to our whole genome sequence data. So then here we'll be building out, and you can kind of see the demographic overview on the right here. We're building out a cohort of participants that have had the condition of breast cancer and also have their whole genome sequences linked. So that's how we're kind of limiting from everybody into our refined section that we want to focus on. In addition to kind of having inclusions, we can also exclude participants. So if we're interested in specifically looking at female breast cancer participants that have whole genome sequences. We can go into this area and exclude based on demographics. We can do exclusion for sex assigned at birth, and we can exclude our male participants. So then this will focus specifically on our female sex assigned at birth, breast cancer conditions with whole genome sequences. From here, if that's what we were interested in, if we didn't want to continue to refine, we could create this cohort, give that cohort a name and a description, and move on from there. I've already kind of generated this cohort, so I'll just move on to the next step. And then we would go back to our data, discard the changes. And once we have developed our cohort, which is here our list of participant IDs, then we're going to move to our data set where we'll actually get the specific data from that cohort to actually do the analysis on. So here we'll have kind of three different main columns. The first column is where we're selecting our cohort. So we can have all participants if we want to look at everybody. But the downside of that is that that is a massive amount of data. So that is going to increase your costs because the cost is going to be based on your computing and the data that you're bringing in. So that's another reason that you really want to select only what you're interested in looking at specifically for your study. So we'll specifically choose our female breast cancer cohort that we generated. And then from here, we can look at different concept sets. So this is where you're going to select the different type of information that you're interested in looking at. So if you're interested specifically in looking at the demographic information of this cohort, you can choose demographics, and then this will show you exactly what values you are actually bringing into your research or notebook. So this is like person IDs, gender, date of birth, race, ethnicity, and everything kind of associated with demographics. If we're interested in linking this data also to different survey data, we can also click that, and then you'll be able to see here under survey, you'll see what type of data we have under the survey concept set. And that's the person IDs, the survey date times, the questions, the answers, and those kind of data associated with the surveys. You can also select and develop your own concept set. So if you're interested in a specific drug or lab and measurement associated with this condition or whatever condition that you're interested in researching, this is where you would go about doing that. So if we were interested in specifically in drug exposure, you can do it similarly where you could be able to search that drug and then find that, add that condition in, and then we'll go back. I'll kind of show an example of that. So if we're doing, let's just say demographics, and we'll have our whole genome sequences linked to that. And then if we're interested potentially in vitamin D levels, we can look at that and have that, and we'll be able to see under these measurements, this is the different kind of data type and the different measurement data that you'll have for vitamin D. All these different data that you'll have associated with that concept set that you built out. So in this case, just kind of reiterate what you'd be doing is that you would have focused on rather than pulling data from all of our participants, specifically the participants that you're interested in looking at. And then here you are focusing on what data you want to be pulling in. So if we're interested specifically in demographic data, genomic data, as well as vitamin D data, that's the data that we're going to be ingesting into our notebook. So then we can create this data set. So we'll say breast cancer, genomic, vitamin D. We can save that data set, and then we'll hit analyze. So I'm not going to go through this process because it's going to take a little bit of time, and then it's much more useful if I kind of show you a built out one. But this is kind of where you would go and extract the data, bring it in. And what this is actually going to do is essentially write a SQL script, which will then interact with our database and pull that into your notebook. So I'm going to skip that. And this is where you'll be actually able to see this code preview. You can either add this code and add this kind of data set to a notebook that you have existing within this workspace, or you can create a new notebook. And then you would also select your programming language at this point. So the two major programming languages that we support are Python and R. For genomic and genetic data, we also have Hale and Plink to help support analyzing that data as well. So I'm just going to cancel that. But from here, once you develop your notebook and pull that data out, then you're going to go to analysis. And then this is where that would show up in a different notebook. So if we want to look specifically at a notebook, I'll show you kind of what a Jupyter notebook looks like. So hopefully it won't take too long to boot up. I tried to start running the environment a little bit earlier. If you're starting up for the first time, it takes about 5 minutes or so. But Jupyter Notebooks is if you're not familiar with it, it's essentially an environment where you are able to write in both normal language, so such as English, as you can see here, as well as coding languages are Python, where you can actually write that into a cell and run that script. So it's great as essentially both a lab notebook where you're able to take notes and really just kind of write out what's happening, as well as essentially a lab workbench where you're able to write your code and actually carry out your experiments analysis. And I was hoping this would go a little bit faster, but essentially what it would look like is different. These are different cells that you can interact with and choose either to write in normal language or in coding language. You can import installed software packages. You can import things from GitHub, whatever you need to kind of be bringing in and working with. A couple limitations is that we do not allow anybody to download our row level data to work with outside of this environment. As I mentioned before, something that we take very seriously is the security and privacy of our participant information. So all the work on this data has to happen on our cloud computing platform. You are allowed to supplement our data and bring in your own data as well. So you can have a combination that way. We just do not allow you to take it out and then supplement your own data with our data, if that makes sense. So essentially what I'm doing right here, which I was hoping was going to go faster, is setting up our Jupyter environment. So you can actually get an estimate on what the cost will be before running your environment and starting everything up. You'll be able to kind of go onto this Thunderbolt, Lightning Bolt tab here. And then this will kind of give you the overview of what it will cost when you're running, when it's paused, and it'll kind of show you the diagnostics for CPUs, RAM. You can actually, now it's kicking in, but you can actually adjust that. That will also adjust your price. But that's one way to adjust the power of your virtual machine that you're working with. And then so now we're bringing up essentially what a Jupyter Notebook environment looks like. So as I mentioned, you have different cells where you're able to write in either normal English or in code, and you can run these cells as well. You can go down and you can actually look at and build out graphs and do your analysis within this environment. So this is really where you're going to be just going. And once you pull that data out, as kind of I showed you in that first section, this is where you're going to have all your data in the secured location and where you'll be able to run your scripts to actually analyze it. To go back to kind of manipulating the environment, you can do that here. Choose your CPUs, RAMs, you can adjust those. That will adjust the price, as I mentioned. And essentially what you actually are paying for is bringing in that data and then analyzing that data. So that's another reason why we highly recommend building out that cohort and not just choose all participants, because then you're working with a much, much, much larger data set rather than only working with what you need to, because we have a huge amount of data and we want to make it as easy for you and as cheap for you as possible. So you don't really necessarily have to worry too much about the storage costs. A lot of that is handled by us. So you're mostly concerned about kind of running the analysis and ingesting that data. So you'll be able to see what that cost estimate is here, as well as what's really cool is that all new kind of researchers that sign up get $300 free credits to start kind of get their feet wet, start working with. And a lot of times, as long as you're not working with genomic data or just absolutely massive data sets, this can actually go quite a long way. After your $300 limit runs out, then you're going to need to be linking to a new billing account. So that kind of keep that going. But that's essentially how you kind of go through figuring out on the public site what data is available, going into the researcher workbench, finding out, okay, how do I get and nail down my cohort, and then develop and pull out specifically that data that I want into this Jupyter notebook setup where I'm able to actually go through and do the analysis. So that's essentially the overview and the process that you'll follow. We do have a number of tools and support systems to really help you through this process. We're not trying to completely let you float on your own. And you can find them kind of under this hamburger menu at the top left. So this is where you'll be able to see your workspaces, but we also have featured workspaces, which are broken down to 3 main categories. So we have tutorial workspaces, demonstration projects, and phenotype libraries. So these kind of serve different functions. So tutorial workspace, what we were actually just looking at in that Jupyter notebook was actually a duplicate of one of these tutorial workspaces. And the idea is to give a blueprint for researchers to look at for different kind of purposes. So how to get started with control tier data, how to get started with registered tier data. So kind of understanding, okay, this is how you kind of ingest and start working with this data, and to more kind of complicated things of how to work with genomic data, specific data types such as the COPE survey, which is linked to our COVID survey. So this is a really, really great resource. If you are interested in using a specific type of data, go to look at these tutorial workspaces, and it'll really show a blueprint of how to go through that. Demonstration projects are similar kind of blueprints, and they show demos of different types of projects that have been done, and specific data types. And then phenotype libraries similarly are also blueprints of how to kind of look at and pull out a specific phenotype. Since there's a lot of ways, you can also, instead of using the cohort builder that we did and dataset builder, you can also write your own SQL query to query our database and pull out data that way. So these are really good resources to look at if you're interested in specific breast cancer, dementia, depression. So a couple of the normal things that you'll pop up or that researchers will want. We have a couple examples there. Finally, or not totally finally, we also have a great kind of responsive team to, if you want to contact, if you have any questions, very, very helpful team. And we also have a user support hub, which is filled like really, really good resources to look at. So if you're stuck anywhere, I know I went through a lot of information very quickly. So if you're like, I remember him covering this, but I don't remember exactly what it is, chances are all that information is going to be in this user support hub. And I see there's a couple icons on chat. I'm not sure what the questions were, but chances are you might have got a link to the user support hub for different articles here. It's very, very useful. Highly, highly recommend just using this the different resources available here. Additionally, there's an event calendar linked here. And here you can see our calendar of different office hours that our support team holds. So we have office hours on Fridays and Tuesdays. Once a month, there is an office hours, which is the new user orientation. Friday office hours tend to have a specific topic that they try to cover, and then it opens up to a kind of free-for-all questions. And Tuesdays is just a complete free-for-all questions. Come in with any questions that you have. If you're fairly new, it can be very intimidating because a lot of times people will have very specific very intense questions, but do not get scared away. If you feel like you have a stupid question or any sort of basic question, that's also a great place to ask. So we want to make this as useful for all of you as possible. And just feel free to use the resources as much as possible. Another thing I do want to highlight very quickly within our featured workspace, if you are pretty new to this process of working with electronic health records or coding or just kind of getting familiar with this program, we have a fairly new tutorial workspace called the Skills Assessment Training Notebook for users. And this will kind of have a couple of different notebooks that will go through the overview of a couple different things that are very basic to hopefully get you more acclimated and a foot in the door, so you can learn the OMOP common data model for understanding how our data is organized, especially the electronic health record data, how to kind of explore that, as well as kind of how to query our data and how to manipulate data for a very, very basic kind of overview. So I know I went through a lot, and I'm sure there's a bunch of questions, but I'm going to end it there and open this up for questions. So if anyone has any questions, feel free to ask. And I think that should pretty much do that.
Thank you, Dr. Lyons. Anybody have any questions that they would like to ask Dr. Lyons or his team?
Hi, hello, Dr. Lyons. My name is Diego Mazzotti. Thank you for your great presentation. It's very nice to see the resource kind of like, pretty much ready to be used there. So I'm hoping that this presentation will really engage a lot of people to be able to use this. So I did have lots of questions. I'll try to kind of like, just go to like a couple of maybe key ones here, or maybe more curiosity, right? So is there a way that you can share, like as you create your cohorts for your study, is there a way that you can share with others, maybe in your team, or do you actually have to, each individual will have to create their own cohorts, and you kind of like share the same protocol that you created your cohort? So is there a way to share?
That is a fantastic question that I did not address. So thank you for bringing that up. We do want to make this a collaborative environment as much as possible. So you are allowed to share your notebooks with other registered users. The only caveat is that they have to be registered users. And if you're using a controlled tier data set with genomics or those 3-digit zip codes, the person that you're sharing with also has to be registered in the control tier. But the way to do that is very, very simple. As I kind of showed, you kind of go to this 3 dots next to your workspace and then just hit the share button, go down to share, and you find your collaborator. So you kind of just type in, so Danielle Wilfong should be in this. She's one of my teammates here. And so if I want to share this notebook with her, I could go in and click there. So it's very, very easy. And we want to make this as collaborative as possible. Yeah, that's a great question.
Okay. And then the other question that I had was about like the billing, right? So is billing only like individual, or can you have like, let's say a group billing that several users can be managed under the same billing account?
Yeah, that's a really good question. So each individual gets the $300 free credits, which is great. So it's not $300 for a group per person. And then from there, when you're actually setting up your billing account, I think if you have a billing account, then I think multiple people can be registered to that account. But I think that one of my colleagues might be able to answer that more specifically if I'm misleading in any way, if anyone wants to jump in.
Yeah, I can take that. It's a great question that we get a lot. The money will come out of the owner's account. So whomever is listed as the owner for the workbench, that's where the funds will come from.
Okay, thank you. And the last one, I promise, I'll let other people ask. So I actually thought the workflow, the process of creating the cohort and coming up with a data set is actually very interesting. It seemed to be like very easy and fast.
Yeah.
Is this workflow like open access, like open source? And is there like a way that we can replicate the same process, for example, work with coming up with data sets for like our own institutional EHR data sets and things like this? Is this kind of like openly available, or it's like specific for all of us? So
the cohort builder is specific for all of us. Because essentially, and all of this, what it essentially is doing is it's using point and click tools to write a SQL script, which will then be plugged in your notebook to actually pull out the data from the database. I don't know how generalizable that script would be to query other databases. Does that answer your question? Yeah, no, assuming like, the data would be in the OMAP data model, for example, I feel like it, technically could work. Maybe not for the other tables that are not like that are beyond what is in the OMAP data model. But yeah, I was just wondering, like, for example, we use here I2B2, right? And then but I do think that this seems so much simpler and so much faster that, I wish we could try to replicate this. Yeah. Yeah, thank you. Yep. If there's any other questions, we're happy to take them.
I have a question, and I haven't read the billing article that was posted in the link, but so if a graduate student wants to use this data for their PhD
Yeah.
dissertation, but we have an account that's affiliated with the institution that we want to bill. How would that work? So we don't want the student to be billed.
Right. So this information should be in that article that was linked
Okay.
for setting up the billing account. But essentially you will have initially, like, each individual will have $300 free credits. So then that will be like your individual, in a way, like, kind of thing that you'll go through. And then from there, you can actually link in whatever billing account that you want to link into. It won't continue to pull, like, negatively specifically. No students can be like putting up their bank account or anything like that. You'll be putting in a specific a kind of a billing system.
Okay.
But yeah, kind of the details of that should all be within that article.
Thank you.
Yeah.
I have a question. First off, thank you for joining us today, Dr. Lyons. I appreciate it. I have kind of been looking into some of this data and thinking about using it, specifically the survey data. And I wanted to know if there was a way to track or know if the same person has taken several of these surveys. You said participation is required when they first sign up, but is it optional later on?
Yeah, so the initial steps into kind of being part of the program and kind of the first data that you're going to have are 3 basic surveys. That's what's going to be filled out. And that's essentially the minimum requirement to have your data kind of go in. From there, you can choose to continue to take more surveys, link your different data types, give a biological specimen sample for genomic data. And in terms of finding out if they have filled out surveys multiple times, I don't think all of our surveys are supposed to be filled out multiple times. I think there's different ones that will be filled out multiple times. There might be a way to do it by building your cohort. And so if we're something like this, let's see if we can do under the criteria selected, if we're able to apply modifiers. Okay, so that's event at age. So I would look into just kind of looking to see if we can find, if you can apply modifiers to look at if they've filled it out multiple times. That would be my recommendation for kind of developing that cohort and finding that participant population. Unless does any of my colleagues have a better idea? And if that also doesn't work, feel free to contact us and ask questions. We're always trying to find out what we can do better. And kind of get that feedback and grow and develop and make it more useful.
Hey, Jennifer, this is Sam from the user support and research support. So for survey data, you can determine by person ID the specific surveys that they have completed. So you can query that information. There is a featured workspace called How to Work with Survey Data, and that will list it out. So once you become a registered user, you should be able to access that. And it'll show you how to pull that survey information. Now, like Michael said, if they only completed the first 3 surveys, they won't have any duplicate survey information, or they should not. So as surveys are updated, which each release, they won't go back and fill out new surveys. They'll just have whatever surveys are available, and that's the curated data repository at the time. And so if you want to indicate which surveys they've completed, would be based on person ID. I will note there's some branching logic within the surveys. And so those branching logics will come up as an individual row within that survey. So there's ways to wrangle that data to where you can put all the answers in a single row, and that will be in that tutorial workspace.
And that's still applicable to the COPE data set?
Yes.
Yeah.
That's
Any of the survey data set will kind of all be handled in a similar fashion. Yeah.
Thank you, guys.
So I think the closest geographic identifier you have is 3-digit zip code.
Yeah.
But that wouldn't necessarily allow us to classify some of our rural counties as to where they are. Are there other types of more generic geographic identifiers in the data set? Is that under the demographics, or how do you address those types of issues? So, for example, if we were particularly interested in some of our rural volunteers and all of us.
That's a good question. So the most accurate that I know of is a 3-digit zip code. I'm not sure if there's a way to like specifically focus on rural versus urban kind of locations. Do any of my colleagues know or can answer that?
I can take that.
I can.
Oh,
Go
sorry,
ahead, Ashley.
Danielle. I saw
Go ahead, Ashley.
I saw all three of us come off mute at the same time. So in addition to like the 3-digit zip codes that Michael was talking about, our surveys, we have a question about rural location. If the participants is in what they consider a rural location, if that helps.
And that would be defined under the survey elements, which we can actually access, right?
Right.
Yeah, and that would be like a self-reported, so it kind of depends on that person.
And I guess the other thing is that we also like to have geographic identifiers that would potentially identify people living in socioeconomically depressed areas. But again, the 3-digit zip code's not going to get us that. So we would have to use the sort of social determinants of health components of the survey, but we can't. So would it all be individual? We can't do any links to geographic components to that.
Right. Yeah.
Yeah, they're so rich. I shouldn't focus on limitations. Sorry.
No. Like I said, we are always trying to make it better and more useful for researchers. So if you have things that you think would be like make this better and more useful and have better utility, like we want to hear that feedback as well.
And we do hope to add that rural location in the future. Once we get more participants into our data set, we have it so that to help to prevent re-identification of participants. So the more participants we have, the more data we have, and the more that we can get, the more granular we can go. So if you're a researcher, we welcome you to also become a participant with us and help grow the program that way as well.
Well, you'd have the ability to create RUCA codes, for example, within the data set and then regenerate those with other derived geographic variables, but we wouldn't be able to do it.
Right. Yeah, we keep all this, we have a lot of the data, and we're hoping to release it with more participants we get.
Yeah, the hesitation for like having very specific is just to prevent re-identification, as Ashley mentioned. So just like a security measure for our participant privacy. So potentially we might be able to get more specific for locations and things like that as our data set grows. But that's kind of our limitation at this point.
And yeah, and that kind of goes hand in hand with our demographics. Well, we've got our race and ethnicity, but we haven't released it just yet in a more granular form. So hopefully one day.
So I saw Samantha's response with the demographic information. Thank you so much. That was very helpful. A remaining question that I have, that resource said, this is obviously not nationally representative, so some caution is needed when you try to generalize. And I'm just kind of curious if there's been much discussion about the ways in which this sample is not nationally representative, or particular ways in which it may be different from the general population. I imagine people who participate are like really motivated to share their data and may look different than other folks in the US. So I'm just curious if there are discussions around ways in which that may affect results, or particular things that we need to have in mind as researchers where we really may not be representing the general population.
Yeah, so that's a really good question. So that kind of brings me back to this slide. We just have a huge amount of participants and community partners that reach out to participants that we're really interested in engaging. So it definitely is a skewed data set in a way of that we are trying to overrepresent specific communities. So that is definitely something to kind of consider and think about. That this is not, as you mentioned, scalable for a general representation of the population in any way. But that's not really necessarily the goal of the program, though. The goal of the program is to have a data set that is different than what historically data sets have been. So you can actually look at specific populations that don't have a whole lot of data and understand that. So for things like our genomics teams and our variants, we're seeing a huge new amount of novel variants that haven't been described previously.
I would also add, and thank you for your comment. I'm going to lean on my my colleague Ashley here, but we do test the data ourselves. So we have demonstration groups who try to see if they can replicate certain things that we know that have been shown within data and with large groups. And so that's one way that we try to ensure that the data that people are using is actually able to say something. I don't know, Ashley, if you want to add anything to that, but it's important that you also know that we're not just putting the data out there, but we're also looking at it ourselves as well.
Right. We have, like what Danielle was mentioning, we have demonstration projects, and Michael was talking about he was showing the tutorial workspaces for demonstration groups, which you're welcome to look at. Before we have a big release, we have volunteers from our consortium that is that big, huge slide that Dr. Lyons showed of all of the organizations we have working with us. But we get volunteer researchers, and they replicate a study that's already been published and peer reviewed. And we do that to show that the data have that they've gone through QA/QC, that they are data that's already been out there that people can look at, and that and we also rely on the researchers when they do this to see if there are any gaps or if there are any problems going on with the data, so that when you're able to get to it, that it's gone through all the processes and making sure that we have everything. So yes, our demo projects, they replicate well-known studies that we have out there. And you can see these on the Research Hub with the publications page.
No, that's somewhere here.
I
Yeah,
put it in
somewhere
the
there.
chat, Michael.
Oh, thank you. Perfect.
All right, well, I think we are a little bit past our designated time, but I really appreciate Dr. Lyons, you and your team taking the time to present this to us today. Really exciting data set, great presentation, really informative. And I also like to thank Brenda and Mariana and the Juntos team for putting this on our radar as well. And really excited for what's to come with this data set and getting involved and doing some work with it and seeing what comes out. All right, if there's no other questions or any comments, appreciate everybody's time and thank you for joining us. Thank you, Dr. Lyons and your team for the presentation.
Yep. Thank you for having us, and good luck.
Thank you.